Assalamualaikum and hi everyone. Today we are going to learn chapter 9, Homeostasis. Here are three subtopics in homeostasis. The first one, concept of homeostasis. Second, negative feedback mechanism. And the last one, human homeostatic organ, that is kidney. So we look for the structure and functions of kidney. For learning outcome, explain the concept of homeostasis and describe the homeostatic control system. Concept of homeostasis. Okay, what is homeostasis? So homeostasis is a process maintaining constant internal environment in physiological limit. Example here, body temperature and water content. So what is internal environment? So internal environment refers to the environment surrounding cells, which are interstitial fluid and blood, and also tissues and organs. Example, tissue fluid in mammals. And it is affected by physical factors such as temperature and blood pressure, and also chemical factors such as glucose in blood and blood pH. Physical and chemical factors usually maintained at a balanced level and controlled closely by homeostatic mechanism from sudden changes. So example of homeostatic mechanism are osmoregulations, excretion, and blood glucose level. So what is osmoregulation? Osmoregulation is control of the water balance in the body. And an excretion is the removal of waste products which would become toxic if allowed to remain in the body. Importance of homeostasis is to ensure the optimum internal environment that enables body cells to function effectively, and it also enables organisms to live in different habitats with a wider range of environmental conditions. There are three basic components of a control system. The first one, receptor or sensor. Next, we have control center. And the last one, we have effector. So receptor, it will detect a stimulus and send information to control center via efferent pathway. And control center will analyze the information to stimulate the action that will correct the changes and transmit the output to effector via efferent pathway, and then effector that receive from control center, so it will carries out the action that restore condition back to normal through feedback mechanism. Okay, many biological processes are self-regulating. It is operated by a mechanism called feedback mechanism. So here we have two types of feedback mechanism. The first one is negative feedback, and next we have positive feedback. So negative feedback, it will reduce or stop the intensity of original stimulus, and then for positive feedback, it will increase or accelerates receptors action from original stimuli. Okay, for feedback mechanism, from the normal level, so if the level increase and then the level keeps increasing, okay, it is called positive feedback. It same goes to when the level decrease and the level keeps decreasing, it is also called positive feedback. But if the level increase or level decrease and correctional mechanism happen back to normal level, it is called negative feedback. Example of negative feedback that reduce the intensity of original stimulus, it includes the regulation of blood glucose level, and it also regulation of body temperature. For example of positive feedback that enhance the intensity of original stimulus, so it is the secretion of oxytocin during childbirth. Okay, next we look for negative feedback mechanism in controlling blood glucose level. Normal blood glucose level in human is about 90 mg per 100 ml of blood. Blood glucose level is regulated by islet of Langerhans in pancreas. So islet of Langerhans has alpha cell that secrete glucagon and beta cells that secrete insulin. So here this is the pancreas that secrete insulin in response to glucose levels in the blood. Insulin and glucagon are antagonistic hormones that regulate blood glucose level. Okay, here that is the pancreas, and pancreas has islet of Langerhans that have beta cells and alpha cells. So beta cells will secrete insulin and alpha cells will secrete glucagon. So please take note, beta cells not B cells and alpha cells not A cells. Two conditions in human body related to blood glucose level in blood. So the first one is hyperglycemia. Hyperglycemia means rise in blood glucose level, example after meal. So beta cells of islet of Langerhans in the pancreas will secrete insulin, and insulin will stimulate uptake of glucose from the blood into muscle cell, liver and adipose tissue. So glucose is converted into glycogen in muscle cells and liver. That is called glycogenesis. Glycogen is stored in the liver and muscle cells. So blood glucose then decrease to normal level. And second one is hypoglycemia. Means low blood glucose level, example after skipping a meal. So alpha cell of islet of Langerhans in the pancreas will secrete glucagon. So glucagon stimulates the conversion of glycogen to glucose. That is called glycogenolysis. And it also converts amino acids, fatty acids, and other metabolites to glucose. That is called gluconeogenesis. So glucose level will increase and bring the glucose level to normal, so it involves negative feedback mechanism. So when glucose level in the blood increase, beta cells will secrete insulin and glucose will convert into glycogen. And when glucose level in the blood decrease, so alpha cell will secrete glucagon, so glycogen will break down to glucose. For learning outcome, next we look for the structure of nephron. Kidney is homeostatic organ that is the major excretory and osmoregulatory organ of mammals. Function of kidney first is excretion, means the removal of waste products of metabolism, example urea, uric acid by formation of urine. And then next, osmoregulation, that is regulate water content. And other than that, function of kidney in regulation of pH, pressure and volume of body fluids. And last one, it involves regulation of the chemical composition of body fluids. Okay, kidney is dark red color, and then kidney consists three distinct regions. The first is renal cortex, that is outer region. Next we have renal medulla, inner region, and renal pelvis, upper part of the ureter. And then here renal cortex, that is outer region, and here renal medulla, inner region. So here that is a nephron, functional unit of kidney. Functional unit of kidney is nephron. So we have about 1 million nephron for each kidney. 6 regions in nephron. So the first one we have renal corpuscle. So consists of glomerulus and Bowman's capsule. Next we have proximal convoluted tubule. And then number 3, descending limb, loop of Henle. Okay, here descending limb, and then ascending limb, loop of Henle. Number 5, distal convoluted tubule, and last one, collecting duct. So first we look for renal corpuscle, that is Malpighian body, where the blood plasma is filtered. It contains glomerulus. So here that is a glomerulus, glomerular capillary, and then here the Bowman's capsule. Bowman's capsule, so each nephron begins as a hollow cup shaped in cortex of the kidney. So renal cortex, here we have Bowman's capsule. Okay, here that is glomerulus, that is highly coiled and surrounded by the Bowman's capsule. So glomerulus, that is a capillary network of one layer of endothelial cell with pore, function in filtering unit in ultrafiltration process. Next, proximal convoluted tubule. So it close to Bowman's capsule and highly coiled. It located in the cortex of the kidney, function in reabsorption and secretion of selective substance. In proximal convoluted tubule, cuboidal epithelial cells with lots of mitochondria, and the inner surface is lined with microvilli. Okay, next we look for loop of Henle. So it composes descending limb and ascending limb. Hairpin shape. So here hairpin shape located in the medulla of the kidney. So has thin descending limb, and thick ascending limb. So function in reabsorption of selective substance. Okay, distal convoluted tubule, that is away from glomerular capsule, located in the cortex. So it composed of cuboidal epithelial cells. The inner surface is lined with few microvilli and mitochondria. So function distal convoluted tubule in reabsorption and secretion of selective substance. Okay, last part of nephron, that is collecting duct. End of nephron. It located in the renal cortex and extend into medulla of the kidney. So function in reabsorption of selective substance. Blood vessel through nephron. Blood enters the kidney by the renal artery from aorta, and it supplies both oxygenated and unfiltered, means dirty blood, that is branches into efferent arteriole. Each nephron receives one efferent arteriole, and it divides into a coil, ball-shaped capillary network called glomerulus. Efferent arteriole is larger diameter compared to efferent arterioles. So glomerulus, that is a network of capillary enclosed by Bowman's capsule. That is a site for ultrafiltration of blood due to its porous capillary. Blood leaves glomerulus into an efferent arteriole that is narrower than efferent arteriole. Efferent arteriole branches into peritubular capillaries that surround the loop of Henle that is called vasa recta. Blood leaves the kidney through renal vein to vena cava and draining out the oxygenated and clean blood. That's all.